Welcome everyone to today's webcast. I hope your week has been good to you so far. I'm Cheryl Rogers, the Director of Marketing at Golden Helix, and today I'm happy to introduce you to Hawa Yusuf, a field application scientist here. Hawa will be discussing using clinical reports as part of a gene panel pipeline. Hawa?
Thank you, Cheryl, for the introduction, and good morning, everybody. Thank you for taking the time to join us today.
Muted.
As Cheryl stated, my name is Hawa, and I'm a field application scientist here at Golden Helix. If you have any questions during today's presentation, please feel free to enter them at any time in the questions pane in your GoToWebinar window. We will try to answer as many of them as possible. If there are any questions we don't get to, we will follow up with you with the answers as soon as possible. So here's our agenda for today. For those of you who may not be familiar with Golden Helix, I will take a minute to talk about us and what we do. And for those of you who are familiar, I promise to keep it short. I will then talk briefly about VS Pipeline, then spend the rest of our time today looking at VarSeq, VS Reports, and VS Pipeline to see how you can use VS Pipeline to automate VarSeq projects from data import all the way to report generation. And as promised, we'll wrap up with a Q&A session. So with that, here's a little bit about us. Golden Helix was founded in 1998, about 18 years ago now. Our founders spun off early work at GlaxoSmithKline, and GSK was a key investor in the company. Golden Helix started out creating products for genomics and bioinformatics with Helix Tree, which has now evolved to be our SNP and variation suite. SVS is a research-focused product that allows researchers to perform genome-wide association studies, genomic prediction, large N population studies, and many other research-oriented analysis. On the clinical side, we have our second product, VarSeq, which allows clinicians to annotate and filter down samples to variants of interest, create clinical reports modeled off the ACMG guidelines, and for our high-throughput users, VarSeq has the ability to run all the workflows via our high-performance pipeline, which we will be exploring today. Finally, VarSeq supports VS Warehouse, which is our data warehousing solution. We are in business with over 350 client organizations, and they fall into 5 different categories. We work with large hospitals like SickKids, CHOP, and others, and many research organizations like Stanford, UCLA, and Yale. Our customers also include labs that fall in the testing lab category, like Prevention Genetic and NCI centers. And we work with major pharmaceutical companies like Bayer and Teva. Last but not least, we work with government organizations, and here in the United States, that includes organizations such as the NIH, the USDA, and the Food and Drug Administration. Within these organizations, we are approaching 10,000 actual users. So some of our clients use our software to get published, and we have over 1,000 citations in publications such as Science, Nature, Nature Genetics, and many more. We are very proud of the fact that when you buy a Golden Helix product, you are getting way more than just a piece of software. We have a reputation for quality products, excellent customer service, and domain expertise. We support our customers every step of the way and engage with our customers to help us understand their use cases and their ecosystems. We keep very active in the community by speaking at, sponsoring, or attending events and conferences. We publish ebooks and best practices and stay engaged within the community. So far this year, we attended ACMG in March and ESHG in Barcelona this last May. Last but not least, we are transparent about our implementation. You can review our methods and citations and externally vet the methods we have embedded in our product. We pride ourselves in speed and innovation and offer highly customizable products. So if you ever have to run the same workflows multiple times, VarSeq is a great tool because it was built from the ground up to allow you to define workflows and run them repeatedly for each sample or batch of samples. First, we create a project which we call the design project. In this project, we define everything about the workflow. This includes defining the annotations and the versions of those annotations that we include, setting up our filters that we use to narrow down to a list of candidate variants, and the parameters that are tuned to our secondary pipeline. We include the fields that we want to display and even set up our clinical report templates. We define all the preferences and the parameters that help us in streamlining the interpretation processes. We then save this design project as a project template. So when we receive new data, whether it be single samples or a batch of samples, we can create new projects with a template and import the data. All of our steps from the workflows are then ran, and so then we can go through each sample, generating our own interpretation, and often running exports, whether it be saving to Excel, VCF, or CSV file formats. VS Pipeline allows you to take a template, build and automate the input of the data into the project, and export of the data from that project. This means importing data into a project and exporting a filtered and annotation variant list into Excel, VCF, or text file formats that could then be passed on to a colleague or other clinicians for further interpretation and analysis. Today, I will be showing you how you can even take it one step further to generate a clinical report within VS Pipeline. These VarSeq projects created by VS Pipeline can be opened in a fully licensed copy of VarSeq or even the free version of VarSeq in viewer mode. The ability to have all these steps automated simplifies the process of putting this in a CAP or CLIA certified environment. This automated process reduces human error and increases efficiency in your lab. Of course, all these steps are logged, so you'll be able to tell which versions of the template was used, the annotation sources and the versions that were used, and when the steps were performed. With that, we'll go ahead and jump into the demo. For today's webcast, we will be using data from the Illumina MiniSeq TrueSight Cancer Panel. We will be looking for variants that are associated with lymphoma, specifically those whose function in cancer includes oncogenes suppressor genes. All right, so here I've got VarSeq opened, and this is the project that we'll be looking at today. As we can see, we've got our 438 variants imported into this project. And on the table here on the right, we can see a list of all 438 variants that we have in this project, along with the annotation sources that we've added. On the left here, we can see our filter chain that we've added for this project. As we can see, I've added a quality control filter that narrows down our list of variants from 438 to 259 variants. I then added a filter card to select variants whose effect is either considered loss of function or missense, and that got us down to 54 variants. Next, I added a filter card to select variants that were present at Cosmic, and that brought us down to 29 variants. Next, I added a filter card based off of our full-rank gene ranking algorithm to select variants that were associated with the lymphoma, and that brought us down to 25 variants. Now at this point, I went ahead and split our filter chain into two sections. On the left here, we've got a filter chain for selecting variants whose function in cancer contains oncogene. And as we can see, that brought us down to 3 variants. We can go ahead and set these variants as part of our oncogene variant set. And to do that, we can just go ahead and click on the square icon and add the current variants to your oncogene variant set. As we can see, went ahead and automatically flagged those for us. So similarly, in this parallel filter chain, I selected variants whose function in cancer contain tumor suppressor genes. And that brought us down to 7 variants. Again, we can go ahead and set these 7 variants as part of our tumor suppressor record set by choosing the square icon here at the top and then choosing Add Current Variants to our tumor suppressor record set. All right, so at this point, we've imported our sample, we've added our annotation sources, we've filtered down to a list of variants for oncogenes and tumor suppressor genes. We can go ahead now and create a clinical report for this sample. To do that, I open up Report Template. And as we can see, we have our patient reference and sample information already pulled in. This information was pulled in with a sample info file that I had specified as part of the import process when I imported these variants. The panel coverage and the average read depth are automatically computed. And then in the section for the patient result, we do not automatically put in the information. This is a section where you would have to go in and enter the result for the patient is either positive or negative. You could then enter your comments, your interpretation summary, and your recommendations for this particular patient. All right, down here we've got our record set variants. So we have our oncogene variants first. We can see it's pulled in the 3 variants that we had flagged as part of our oncogene record set. And we can see that in the interpretation field here, it's pulled in information from OMIM. Similarly, for a tumor suppressor variant set, we had imported those 7 variants that we had set, and it also brought in the information from OMIM. The information in here can be edited. You could add more to it. You could take sections away from it, but it serves as a good template to get you started up and running as quickly as possible. So if we go ahead and render this report template, we can pull this up to see what it looks like. We can see that it's brought in our information into a stylized report. So up here at the top, again, we have our lab information, our physician information, patient and sample information. We can see our results. Here we have a table of our oncogene variants as well as our tumor suppressor variants. Down here we have information from OncoMD containing drugs that target this mutation. And it also has all the information that we had for each variant coming in from OMIM. All right, down on the bottom here, we can see that it says draft report, and we'll take a look at that later in this webcast. So at this point, we've set up our workflow just like we want. We have the annotation sources that we want. We have our filter chain set up quite right. We also set up the conditions for flagging our different variants for the oncogenes and for the tumor suppressors. So we can go ahead and save this project as a template so we can use it in future projects as a template in VS Pipeline. So to do that, I just go ahead and click on File, and I choose Save Project as Template. It brings up a dialog that asks us for a template name, a description, and some more information about that particular template. We can go ahead and choose Save, and then we can use this template in VS Pipeline. So I'll go ahead and switch my screen over to show you how we do that in VS Pipeline. All right, so here on the right, you can see that I've got our VS Pipeline command shell open. And on the left here, we have our batch script that we run in VS Pipeline. So how this works essentially is that we create a script full of commands that we want to perform in VS Pipeline, and then we run that particular script in our command shell for VS Pipeline. Just to give you a quick overview of what this particular script does, it goes ahead and creates and opens a project in VarSeq for us. it automatically imports our VCF files, adds our annotation sources, and then runs through our workflow, like setting up the filter chain, narrowing down our list of variants to just those of interest, as well as flagging those variants. We then export our result into an Excel text in a VCF file, and as well as set up and render a clinical report for the sample. Finally, we just go ahead and save and close the project. So I'll switch over to my VS Pipeline command shell here and go ahead and run this particular script. As you can see, it's loading in the batch file that we specified. And it's importing our variants, setting up our resources. It's going to start annotating our variants against those that were set in our template, and run through everything that we have in our workflow. While it's doing that, I'll go ahead and open up the folder in which we are saving this particular project into. Okay, so here on my screen is the folder where we have the project going into. We can see that we've got this particular project. We can open it and see that it's already created our VS project for us. It's imported all our data. It's also starting to create our report. And while that runs, I'll go ahead and open up a previous project that we'd already ran prior to this webcast, just to save a little bit on time. So here are the results of that project. Again, we see that we have our project that's already been created. We also have the results of our Excel, VCF, and CSV exports. We can go ahead and open up any one of these. And here we see that we've got the 10 variants that we had narrowed down to. The 3 that were found in the oncogene were flagged as true in this Excel spreadsheet. And then the 7 that we had found as tumor suppressor genes were flagged as true in the tumor suppressor column. It's also brought in our annotation sources like VarSeq, and the other annotation sources that we've added to this particular project. Okay, so we can also see that in this folder it's created our report for us. If we open up the report folder, we can take a look at the report that has been generated by the commands we ran in VS Pipeline. So I'll go ahead and open up this. And I'll switch my screen over. And here we can see a report that's very similar to the one we saw when we were first creating the workflow. Again, it has our lab and patient information and our sample information as well. It's got our oncogene variant set, as well as a tumor suppressor variant set, and the drug information for this particular sample. Down here at the bottom, again, we see that it has been approved by me. Well, we can go ahead and change any of the information here in this particular report template by opening up the report or by opening up the project in VarSeq. So to do that, I'll just go ahead and launch a new instance of VarSeq. We'll give it a minute to open that up. And of course, it popped it on my other screen. So we can go ahead and open up the project that was created by our VS Pipeline in VarSeq to take a look at that report and add our interpretation, comments, and recommendations. So to do that, I'll just go ahead and open the project here, locate the save location for this particular project. Okay, and I believe it was this project. And we'll give it a minute to open up. All right, so here's the project that was created by our VS Pipeline commands. Again, we can see that it's, created our filter chain just like we had set in our project template. It's also ran our variants through the filters. It came out with 3 for variants that had function in cancer that contained oncogene, as well as the 7 for variants that had function in cancer that contained tumor suppressor genes. Here we can also see that it's went ahead and automatically flagged our variants for us, the 3 for those oncogenes and the 7 for tumor suppressors. Down here at the bottom, we can also see that it's created a report view for us, complete with our lab and patient information, as well as the variant information. So if we go into our report template, we can go in and add our interpretations in here. Now just edit this a little bit just so we can see a difference. Okay, and then we can go ahead and once again sign off on this particular report by clicking on this verify button, and then we can go ahead and re-render the report. All right, so looking at this report, it should look identical to what we just seen, except for this time it should have edited that interpretation summary field as well as the recommendation fields. And then at the bottom, it also has my approved stamp. You know, it has my name, it was approved by me, and on this date that it was approved. So from here, we can go ahead and export this report as a PDF so we can share it with, or easily share it with other colleagues or clinicians. So that brings us to the end of our demo portion of this webcast. I showed you how to set up your workflow in VarSeq, save that as a project template, and use that in VS Pipeline to automate import of samples and analysis of those samples to generate either Excel, VCF, or text files, as well as clinical reports. If you have any questions in this time, please enter them in the GoToWebinar pane. Or if you would like to request an evaluation, you can reach us at info@goldenhelix.com or visit our site at goldenhelix.com. Thanks so much for tuning in, and Cheryl, I'll pass it back to you.
Thank you, Hawa. As Hawa mentioned, we will answer some questions now, so go ahead and enter those in the question pane at this time. A couple of quick announcements from me. The webcast recording and slides will be up on our website tomorrow, Friday at the very latest, but I will be emailing those to all of you, so look for those in your inbox. We will have our next webcast presentation on Wednesday, August 10th, and that will be presented by Dr. Rowan Palmer of Brown University. Dr. Palmer will be presenting his abstract from our abstract competition this past winter. His abstract is titled Investigating Shared Additive Genetic Variation for Alcohol Dependence Across Individuals of African and European Ancestry. You will see those invites in a couple of weeks in your inbox. Last but certainly not least, we recently launched an e-commerce store, store.goldenhelix.com, and there you can see that we've put together some special packages for our customers where you can quickly and easily buy Golden Helix software products, as well as set up monthly payments, which have been really useful in getting people up and running quickly. That's all for me, so let's answer some questions. First question, can the pipeline handle multiple samples, such as batches of 10 or 20?
Absolutely. VS Pipeline can handle batches of samples. We have a special command for importing samples that come in from, let's say you have a folder that had a lot of different samples in it and you wanted to run all those samples through your workflow. We do have a command that allows you to do that. It creates separate projects as well as export the text files, the VCF and Excel files, as well as create reports for those samples.
How can you use the pipeline with tumor normal or trio analysis workflows?
So you would use the pipeline similarly to how we've done today with trio or two more normal samples, you would set up your workflow in VarSeq, just like that, except for instead of setting those particular filters that we did, your filters might be a little bit different. You would bring in your sample files just like we did, and then you'd save that as a project template, and then you'd use that project template in your pipeline batch script.
Can reports in VarSeq be customized?
Absolutely. So reports in VarSeq are highly customizable. With a little JavaScript and HTML know-how, you can customize the report to your heart's content. In fact, I have a couple of reports that I can show you just to give you a feel for what they look like or how you can really make them different. So this particular report template that we're looking at is for a single sample or single exomes. You can see that it looks completely different from the one that we showed in the webcast today. It's got a different style for it. We've got lots of colors going on for it. Our tables look very different. Even our sign-off section here at the bottom looks different. You can put in your CLIA ID if you have that. If you wanted sections of the report to be omitted, or if you wanted to add new sections or different kinds of information, you could do that with a report template. If you do not have the skills in-house to customize these report templates, we will be more than happy to help you with that. And here's another example of a report template that looks really different from the two that we've seen. Again, this has more of a text feel to it. We wanted to kind of keep this one a little bit simple, but also very interactive, right? So we can collapse sections and display them as we wish, just to kind of keep it a little bit more clean. You can certainly do that. So again, with just a little bit of HTML and JavaScript know-how, your reports can be as customizable as you want or need them to be.
Last question here before we wrap up. Can the pipeline be run on all platforms?
That's a good question. So the pipeline can be run on Windows and Linux platforms. We currently do not support the Mac platform. I know I was showing you my projects here opened up on my Mac, but I do want to let you know that I was running the pipeline commands on the Windows side of my computer.
Thank you, Hawa. Thanks everyone for attending, and thanks to you, Hawa, for a great presentation. We're going to go ahead and wrap it up for today.